Thank you. Hi, everyone. I'm Caio Seguin. I am a research fellow at the Department of Psychiatry at University of Melbourne. So for today, I'm going to talk about neuroanatomical pathways of TMS therapy for depression. And I think the plan will be to start with laying out the modeling framework that I'm going to use for the talk, and then we'll move on to the application for TMS therapy for depression. But before I do that, I would like to show you this. So this is a network representation of the human connectome, where the nodes represent gray matter regions and the links represent white matter connections. And I think the whole enterprise of network neuroscience, what it tries to do is it tries to abstract the brain, this very complex biological organ, into this simpler representation, this network of nodes and edges. And what we gain from this abstraction is that we can now use tools from maths, from physics, from computer science, and from engineering to learn about the structure of this network. But what I think is really important, and it's the theme of this symposium, is that it is crucial that we get what we learn in this level of abstraction and translate it back to biological and clinical insight about brain function. So today I'm going to talk about network communication models. And I like to start with this little example. So here we have a schematic of the human connectome. It's the same as before. The nodes are gray matter regions. The edges are white matter connections. And I've highlighted two regions here of interest in orange, I and J. And we can think of these two regions, they have some sort of functional coupling, functional connectivity, perhaps in a particular behavioral context. But interestingly, they don't have a direct white matter connection between them. So communication between them has to be mediated by a multi-step or polysynaptic path through this network. And the idea of the types of models that I'm going to talk about is that they want to use concepts from graph theory and network neuroscience to model neural signaling through this network. So here I'm showing the shortest path, a path, and a walk between two regions. So how does this work? So another way to think about it is that we want to quantify the structural capacity for communication in the connectome. So here we have a structural connectivity matrix, which is typically sparse because a lot of regions are not directly connected to each other. But if we use a communication model, we can simulate signaling, or can quantify signaling between any two pairs of regions. And if we do that for all region pairs, we get what we call a communication matrix, which is now fully instantiated. So what can we do with this matrix? Another thing that I should mention is that these models are very, very simple models. They are models that favor abstraction over biophysical detail. And I'm going to try to convince you that these models can have good clinical utility. So the first thing, this is a paper we tried to do, we've done a few years ago, is to use these types of models to predict propagation of direct electrical stimulation. This is what Marilyn talked about in her talks. She gave a very good explanation of this paper, probably better than what I will do now. So what we did is we used this big data set of intracranial EEG, and we were interested in this question of when you stimulate a region and we observe causal response of that stimulation in a downstream region that doesn't have an anatomical connectivity to it, can we predict that propagation through the network using these very simple graph models? I'm not going to get into the detail of the mechanics of these models, but we tested different ones that rely on different conceptualizations of brain-network communication. And what we found is that generally we can predict the propagation of stimulation through the brain based on these approaches. So this is causal signal propagation along polysynaptic pathways. And this provided the foundation to then go on and ask, can these network models be used to guide clinical brain stimulation? So I'm going to talk about TMS therapy for depression. I'm going to try to lay out the context for this project. The person who's taught me everything about this is Robin Cash. I don't know if he's here. He was here before. So if I say something wrong, Robin, it's on you. So brain stimulation is a therapy for drug-resistant depression, so people who don't respond to antidepressants. And the right subgenual cortex, or the subgenual anterior cingulate cortex, is really a key region of interest. in this approach. So if you do deep brain stimulation, like the work of Helen Mayberg, you can put an electrode right around there, and you stimulate that region, and patient seems to get better. What we're going to try to do is we're going to try to, instead of invasively stimulating this region with DBS, we now want to non-invasively stimulate it with TMS. But the interesting thing is that this region is a deep cortical region, so you can't target it directly. So what people do is they deliver TMS to the left DOPFC. And this has been shown to modulate activity in the SGC. This has been shown by intracranial EEG combined with TMS, and also by TMS concurrent with fMRI, so TMS inside the fMRI scanner. Today, I should say, one of the difficulties here of using TMS is that the DOPFC is this very large region, right? Shown there in panel B. So there is a problem of where exactly you target your TMS. And today this is done based on functional connectivity. So this is the work of Robin, who is there. And there's this very interesting finding that is super robust and is being replicated by many groups, which is if you actually deliver your TMS to loci in the DOPFC that are more anti-correlated to the SGC, people tend to respond to treatment better. So you want to target places of anti-correlation with SGC. This is a pretty interesting thing. But what is even more interesting from my perspective is that there is no anatomical connection from anywhere in the DOPFC to the SGC. This is based on the neuroimaging tractography that we have done, but it's also corroborated by track tracing studies in monkeys. So because of that, this downstream modulation of the SGC is thought to be polysynaptic, so mediated by a path through the connectome. And as of today, these paths are unknown, and this kind of prevents advancing the treatment and a mechanistic understanding of how this treatment works. So can these network models explain treatment outcome of TMS in depression? So we started with this cohort, which was publicly available. It's 25 patients. They receive repetitive TMS to the left DOPFC. So here in this diagram, every circle is a brain region in our pacillation. The blue dots are the TMS stimulation sites in the DOPFC, and the red dot is the SGC seed. So it's a sort of SGC sphere. And what we want to do is we want to model communication from the TMS sites to the SGC via white matter connectivity. To do that, we used the normative structural connectome. We actually didn't have diffusion MRI data for these patients, so everything's done on a normative structural connectome. And variation across patients just comes from the variation in the coordinates of where the TMS was delivered to. And this is what we found. So here in the x-axis, we have the number of white matter hops from the stimulation sites in the DOPFC to the SGC. I'm going to talk about the modeling in a second. But you can think about how many steps you need to take. And on the y-axis, we have the treatment response. So more is better here. So this is the difference in the MADRS scale before and after treatment. So what we see here is that patients stimulated at DOPFC sites connected to the SGC via shorter communication routes, so more direct, more efficient communication routes via the connectome, experience greater symptom remission. And then we went on and we replicated this in an independent cohort of 24 participants. So these are different clinic in another country, different TMS, different everything. And we found the same results, which was pretty cool. And we also replicated it using a different normative connectome data sets. So for people interested in the particulars of the model, it's quite interesting because we tested a lot of different conceptualizations of network communication, shortest paths, diffusion, communicability, search information. And I wrote maybe 4 or 5 papers saying that you shouldn't do shortest paths because diffusion models, communicability is better. But what worked here was the shortest path. So this basically is the number of hops. On the x-axis, there's the number of hops from the DOPFC to the SGC along the shortest path. So in this case, it was okay. What was cool about the shortest path is that it provides a really simple way to actually go into the model and dissect what are the anatomical pathways that take you from the stimulation sites in the DOPFC to the SGC. I think the colors are good. Yeah, so in dark blue, we have the stimulation sites. In red, we have the regions in the SGC seed. And in purple, we have the mediators. And what is interesting, so this is a kind of a sanity check. If you plot the number of hops from the stimulation sites to the SGC sites, you see that in the shortest possible way to get between these two sets of regions, you need at least 2 hops. And this is because, as we saw, there's no direct anatomical connection between them. So what were the key mediator regions and the key paths between the DOPFC and the SGC? So the key region was the thalamus. This big thing here. Sorry, my hands are shaky. Can't be a neurosurgeon. So the thalamus was very important. And then you have a series of kind of superior frontal regions, kind of along, oh my goodness, where did it go? Superior frontal regions kind of along here. And then if you look at the paths, the main path was this cortical thalamocortical path, where you go to superior frontal region, then you descend into the thalamus, then you come back. And then the second most important one was a cortical path, which looks like this. Sorry, I should have mentioned this. It's my bad. So actually, the one thing that is really interesting is that the stimulation is delivered in the left DOPFC, but the SGC seed is actually on the right hemisphere. So all of these paths are interhemispheric. They have to cross at some point. I always forget to say that. So we stimulate in the left DOPFC, and in this cortical path here, we first go to more or less homotopic regions on the right, superior frontal. Then we go down to sort of an anterior cingulate region, and then we reach the SGC. And I plotted the cortical path because that was the one the reviewer liked. So how does this advance the current state of the art in TMS guided, in fMRI guided TMS? So as I said before, today, TMS targeting is done using fMRI. And it's this idea that you want to stimulate on sites of negative DOPFC, SGC functional connectivity. So we thought, okay, how does this compare if we now do a map of DOPFC, SGC white matter hops? And we compare that to the DOPFC, SGC functional connectivity. And we saw this really nice correlation saying that the sites that are most anti-correlated, so the ones that you target using fMRI, are also the ones that have the shortest communication routes, the ones that you can target based on white matter connectivity in a polysynaptic pathway. So I think this is cool because it provides a sort of a neuroanatomical explanation as to why fMRI-guided TMS for depression works like this. Okay, so this work is published in this preprint, and I'll leave that there while I go through some takeaways. So network communication allows the mapping of polysynaptic white matter pathways, and this potentiates the study of communication between regions that don't have a direct connection between them, which I think is really cool, so you can start to unveil these kind of multi-step circuits. This predicts patient outcomes in TMS therapy in 2 cohorts, and it provides a neuroanatomical grounding for fMRI-guided brain stimulation. And the last thought that I want to say is that this is not, even though it's a network controllability symposium, this was not a talk on controllability. But I think I actually really like that because I think these kind of silos between modeling domains are not very productive. And every model, even if it's a very simple graph theoretical model, has something to contribute, whereas super mechanistic biophysical models also have something to contribute. And I'll end with acknowledgements. Everyone in the author list has helped me a lot. As I said, Robin, Cash, Andrew, Luca provided data, Paul Fitzgerald provided data, Rick provided a vibe, it was really good. Sina made the visualizations. Everyone did, everyone helped. And with that, I will end here. Thank you very much.